Hello, everyone. Thank you so much for taking the time to watch my poster presentation. My name is Melissa Reardon, and I'm from the Department of Chemistry and Biochemistry here at UTA. Today, I'll be talking about charge effects of Ruthenium2 complexes on microtubule stabilization activity. First, I want to talk about Ruthenium2 Polyperitol complexes. Ruthenium2 Polyperitol complexes have been found to be potent or cytotoxic, as well as have acceptable tolerance in animals. These two factors makes them very promising as potential anti-cancer therapeutics. Until recently, these RPCs have actually thought to be targeting the nucleus and or the mitochondria. In 2020, our lab actually found that this complex over here, Rutris dip, accumulated mostly within the cytoskeleton. Now, this is again very interesting because, like I said, until recently, we thought that these RPCs were targeting the nucleus and the mitochondria. And actually, this particular complex was thought to be targeting the mitochondria. So, this is super cool. So, what is in the cytoskeleton? The cytoskeleton is made up of three different types of protein fibers: microfilaments, intermediate filaments, as well as microtubules. Now, microtubule is the most abundant cellular protein and is made up of alpha and beta tubulin heterodimers. These heterodimers come together to polymerize and form this microtubule. When this microtubule breaks down, it is called depolymerization, and it breaks down back into just these two alpha and beta tubulin heterodimers. Now, this polymerization and depolymerization readily happens in the body based off of cellular function, shape, and replication. Scientists actually found that they can alter this stabilization and destabilization of these microtubules with additives. And these additives are called microtubule targeting agents. Now, to this day, all of them are organic based, and they take about 40 or more steps to synthesize. Or if they are natural based, you have to extract them. And as you may imagine, if we widely use this for anti-cancer treatment, we are going to probably extinct these plants. Scientists have also worked on trying to partially extract and then synthesize the rest of the organic complex, but this still takes a lot of steps, time, and money. So it's very fascinating that we found a complex that not only is cytotoxic, but targets these promising targets, microtubules. So we've been investigating these +2 RPC complexes, and they have been shown to actually still interact with microtubule dynamics. Now, Ruthenium polyperitol complexes can be changed to cyclometallated complexes, which means that instead of having this nitrogen group here, you change one to a carbon. Now, different people have shown that this can actually enhance the cytotoxicity. Therefore, I want to see if I change one of my family's complexes to have one of their nitrogen groups to a carbon group, if this can enhance the cytotoxicity of my most promising complexes, as well as still target my microtubule. Now, our complexes are also very interesting because, as you can see on the bottom, they do not take a lot of steps to make. And this is actually the longer steps for cyclometallated. My RPCs appear to take even less. So this is the family of complexes that I'm proposing to make for my cyclometallated complexes. And I have indicated here at the bottom that I've already synthesized and characterized this one. Once I have synthesized and characterized all four complexes, I will be going ahead and testing their cytotoxicity. I want to see how cytotoxic they are against cancer cells. I'll be doing an MTT to do this, and complexes that are less than 10 micromolar will be considered promising. I'll also be investigating their solubility using log P, a shake flask method that determines partition coefficient between octanol and water. The most promising complexes will be investigated for their uptake as well as their subcellular localization, in hopes that they target also mainly the cytoskeleton. Complexes that are promising will be investigated to see the effect they have on microtubule dynamics, so that polymerization and depolymerization I was talking about, as well as any morphological effects on the actual cell. Now, these complexes that I'm talking about actually have enantiomers, which means they have right and left hands. Now, it has actually been shown that some enantiomers can be more cytotoxic than others, as well as cause unwanted effects. Therefore, the FDA actually requires that you investigate these. Therefore, the most promising complexes will have resolved enantiomers and be further investigated. Thank you so much again for taking the time to listen to my poster presentation, and I hope you all have a great day.